let's talk about flowers. Now, flowers are really interesting. They're just modified leaves, but they look really dramatic. And it might take us a little while to remove the color, remove the drama, and just see a flower as being modified leaves. Most of the time a plant is going to be using leaves for photosynthesis. Those leaves have lots of surface area, lots of chloroplast organelles, lots of photosynthesis, making glucose, making energy that the rest of the plant can use. So a big question is, why would you dedicate some of your leaf structures? Why would you put in the energy towards making something that's not going to generate any food? And it turns out that flowers are all about sexual reproduction. Now, the group of plants that produce flowers are called angiosperms. We'll learn later this semester about the other divisions of plants, the mosses, the ferns, the gymnosperms. We'll learn about those other plants that are doing sexual reproduction, but they're not dedicating or making this intense flower structure. So our angiosperms are one of the newest groups of plants on our planet, and they are doing this unusual thing of making a flower structure, of taking some of their leaves that should otherwise be making food and dedicating those leaves or allowing those leaves to have the sole function of sexual reproduction. So when we talk about sexual reproduction, the big question is why? Why go through all this effort? Why invest all of this energy? Why do a specialized type of cell division? Why do sexual reproduction? And it's all about mixing up the genes. Now, consistently in this class, I have tried to emphasize the times or the places or situations where we have seen a loss of genetic variation, that genetic erosion. And when we lose genetic variation for that species of plants or species of any organism on this planet, for that group of species, individuals as part of the species, they have lost the flexibility to adjust to a changing environment. And the environment is always changing. So you want genetic variation. Genetic variation can give you the possibility to survive when the environment changes. So this is why we see plants and other organisms on this planet invest so much energy in sexual reproduction. It's all about giving hope for the future. Hope that future generations, future individuals will be able to survive when the environment changes. So let's get into the details of a flower. Now, what I love about flowers is that even though they have a lot of different ways that they can look, there tends to be some consistency. So we have a nice rule about flowers. There are 4 whorls, and we have to stay rooted in these 4 whorls. We have to keep coming back to it because it is the consistency that we see in how flowers are made. So our 4 main whorls are going to be the sepals, petals, stamen, and carpels. Now, whenever we look at the 4 whorls, we need to pretend that we are looking straight down on a flower, and we're going from the outside in. So our outermost layer will be our sepals. And so down here I'm going to draw a little picture as if I were looking straight down on a flower. And I'm just going to draw my whorls as straight lines. And I'm going to do 3 straight lines. So here for my sepal whorl, I have my 3 main lines. So it's my outermost whorl. If we think about the function of a sepal, the sepal is meant to protect the entire flower. It's meant to protect the entire reproductive structure. Our next layer in will be our petals. And again, I'm only going to draw 3 petals. So here's 1, here's 2, and here's 3. Our next layer in will be the stamen. Our stamen are our male reproductive structures. Our stamen are going to consist of a pollen chamber. Inside of that pollen chamber will be the pollen, the male gamete. It's equivalent to human sperm. And then we'll have the connecting structure that connects that pollen chamber with all the pollen inside back to the rest of the flower. So our next layer will be our stamen. I'm just going to draw 3 little short lines for our stamen. Our innermost layer, our innermost whorl of our flower will be the carpel. The carpel will have our female gametes. It will have the ovary with the ovules, and inside of the ovules, we will have the egg. So I'm going to draw that as a great big circle. And the reason why I'm doing that is because I want you to start to think about this carpel becomes the fruit. It becomes the cucumber. It becomes the green pepper. It becomes the banana, the grape, the avocado. It becomes the pumpkin. And when you think about some of those huge pumpkins that can get hundreds of pounds and be just ginormous, that is one carpel. That is one big ovary with lots of little ovules and lots of little ovaries on the inside. So when we think about a flower, we've got our 4 whorls. Sepals on the outside, petals, stamen, and right in the center will be that carpel layer. Now I wore this rain jacket on purpose because I think sometimes when we're looking at flowers, we tend to forget the whorls and we forget what they do. So the sepals will always be the outermost layer. It's going to be the protective layer, thus the rain jacket. It's protecting me. It's going to protect all of the reproductive structures inside of the flower. Now, for a lot of plants, the sepals are going to be green. And once the flowers mature, the sepals will often fall off. But for a lot of plants, those sepals are going to do double duty. They're going to protect the developing flower, but then once the flower is mature, the sepals will start to express some color. They'll start to look a lot like petals. So our next layer in will be the flashy petals. You can think of that as like the clothing that we wear. A lot of our clothing is very flashy. Sometimes our clothing is meant to attract, it's meant to attract the individuals that we're interested in, but you could think of it as, I'm trying to attract a pollinator. So when we think about petals, petals are often brightly colored. They have intense pigments along the veins, along that vascular tissue, the xylem and phloem bundled together. Those intense pigments can act like runway lights to insects that are seeing the UV, seeing the color underneath the UV. Come on, kitty kitty. Are you going to come into the lecture? All right. Well, let's go talk to the camera. That's a Tohi. He wanted to see what we were up to. I'm sure he's going to chew on some of the flowers. Please remember that lilies and many of the flowers are very toxic to our pets. All right. So the petals have intense pigments that are meant to attract the pollinators, and they're going to see those pigments because they can see under UV. They see using a different light spectrum than what our eyes will necessarily see. Now the next layer in will be the stamen, and I know that we're familiar with humans. So if we think about humans and we think about how humans have their gametes arranged on their body, the male gametes that are often in the testicles. So if we think of the testicles as being like a pollen chamber, and inside the testicles will be sperm, inside of the pollen chamber will be the pollen, and the pollen contains the sperm. Now, like plants and humans, the male gametes tend to be on the outside. So our stamen are going to be exposed. They need to be exposed. We want the pollen, we want the sperm to get transferred from one flower to another, so we can't keep it all hidden on the inside. We have to put the pollen chamber and the pollen out there. We have to get the pollen exposed to the insects or the wind, whatever will be helping move the sperm, move the pollen from one plant to another. Now, in humans, as in plants, the female gametes are contained inside of ovaries, and those ovaries are often deeply protected. So if you think about the human body, the ovaries are deep on the inside of the body, and inside of the ovaries will be, in humans' case, the eggs. Inside of the ovaries of of flower, we're going to have the ovule. And the ovule is a little bit of a different structure than what we see in mammals and humans. So I do want to spend just a quick second talking about the female reproductive structure, the carpel, and specifically that ovule. So if we're looking at our carpel layer, the innermost layer of our flower, our carpel layer is going to have, at the very tip, a stigma. The stigma is going to be sticky. This is where all of our pollen is going to get trapped or will get stuck. And then connecting this stigma to the rest of the ovary, we're going to have the style. Down here, this round bit that I just drew is going to be our ovary. And inside of our ovary, we're going to have ovules. Now, for the sake of the drawing and you being able to see what's going on with the ovule, I'm going to draw it as if this ovary has just one ovule inside. In reality, an ovary is often going to have hundreds of ovules inside. But let's just blow up one of them to see the structure, because structure is really interesting. We're going to have 7 cells with 8 nuclei. So here is my one great big ovule. I'm just going to write ovule up here at the top. And in our ovule, we are going to have, at the very top here, 3 antipodal cells. Well, that here in the middle is a circle. Now, these 3 antipodal cells are not, we don't really know what they do yet. We're still researching and trying to figure out their role. Down here, we are going to have our 2 synergid cells. Now, I'll talk about the synergid cells in just a minute. In between our 2 synergid cells, we're going to have our true egg. The true egg is in red. The remaining structure here is going to be one cell that has 2 nuclei, and these are called the polar nuclei. So one of the things that will happen at fertilization is at fertilization, the sperm will land on the stigma. Now, I'm going to draw my pollen with the sperm inside as a black dot on top of my stigma. We're going to assume that this pollen is of the same species as the rest of this carpel structure. Assuming that it is, then this pollen grain will be allowed to germinate. Let's say that this carpel layer came from a lily that Tohi seems to want to chew on. Let's say this carpel layer came from a lily, but this pollen came from the rose. This happens all the time with flowers. Bees and other pollinators don't care about whose pollen they take to which flower. And so one flower could have lots of different species of pollen on it. And so the plants have to control which pollen is going to fertilize the egg. And so what's going to happen is there will be chemicals secreted by the stigma that will suppress and stop pollen from growing unless it's of the same species. So this is our lily carpel layer. We're going to assume that that is lily pollen and that the stigma is going to allow that pollen grain to start to germinate. Now our pollen grain has 3 separate cells. One cell is going to grow the pollen tube. And the wild thing about pollen tubes is that they've got to grow through the style, they've got to grow through the ovary, and they've got to connect with the ovule, and they've got to connect with the ovule at the correct place so that another sperm can move down and fertilize that egg. So when we look at the ovule, this is where our synergid cells become so important. These synergid cells are going to guide that pollen tube in. It's almost like they're saying, come on, come this way. I used to work at the airport, so we used to marshal in the airplanes. I always think of the synergid cells as being like the marshalers going, nope, nope, a little bit to the right, okay, straight forward, a little bit to the left, all right, coming, and connected. Once that pollen tube connects, both sperm move down. One sperm combines with the egg. As soon as that combination has happened, mitosis, cell division, divide, start to grow and grow and grow and grow and grow. It's going to grow the baby dormant plant. It's going to grow that embryo. Super excited. The other pollen is going to combine with the polar nuclei to make triploid cells with triploid DNA. When we say triploid cells, what we mean is that they have 3 copies of each chromosome. This combined with the polar nuclei combined with the sperm is going to make endosperm. We looked at monocots, the grasses, the wheat and the corn. Most of that seed structure was endosperm. Super nutritious. That is food for the embryo, but we get a hold of it, we eat it, we turn it into bread, we turn it into tortillas and corn chips and all the other delicious stuff that we love to eat. So endosperm, wonderfully delicious, very high in protein, is meant to be food for the embryo. Now in eudicots, like our fast plants, like the legumes, what happens is that embryo will start to immediately consume the endosperm. It will eat all the endosperm and make great big cotyledons. Those are the 2 sides of the seed. When you think about the peanut seed that we dissected, the butter bean, we split those in half. Those 2 great big cotyledons were post embryo consuming the endosperm to make those 2 cotyledons. Once those 2 cotyledons have been made, then the seed goes dormant. And then when it grows again, it can then use that energy in the cotyledons, which is why on your fast plants, your little cotyledons should have fallen off as your plant matured, as your plant consumed all of the energy in those cotyledons. Okay, so we got really into the details of a flower and fertilization. Most plants want to do self-fertilization, but self-fertilization has some drawbacks. If you're just combining your own sperm with your own egg, you're just remixing up your genes. So if I could do self-fertilization, I know that I wouldn't actually have a whole lot of variation in my kids. I've got red hair. Red hair is a recessive trait. This means I have a homozygous recessive genotype. Both pieces of DNA have the recessive gene for red hair. So if my sperm combined with my egg, all we'd get is red-haired kids. There would be no other possibility because that's the only gene that I have. But if you're heterozygous for hair color, maybe one piece of DNA is for brown and the other piece of DNA is for red, well then if you self-fertilized, you could end up with some redhead kids, you could end up with some kids that have brown hair, you could end up with some variation. But it's not a whole lot of variation. You're just reshuffling your own genes. So, kind of boring. Doesn't give you a whole lot of flexibility. If you can combine your gametes with a different plant's gametes, that plant could have different genetics. It could have a recessive trait or a dominant trait that you don't have. You can get that genetic variation that we know makes for a stronger organism, makes you more likely to survive makes the kids more likely to survive when the environment changes. So that's cross-fertilization. We want to be crossing the gametes of multiple different individuals to try to get as much genetic variation as possible. Our last fertilization term that we have to talk about is double fertilization. And this is just referring to the idea that one sperm will combine with the egg to make the embryo, that baby dormant plant. The other egg will combine with the polar nuclei to make the endosperm. So now that we've got the basics of not only the flower, but why sexual reproduction is so important and how the flower helps with sexual reproduction, helps get this genetic variation. Now let's do some flower dissection. Keep that overhead image of a flower in your mind. Sepal, petal, stamen, carpel. keep that layering in mind whenever you're doing a flower dissection. Now, the one thing that I want to point out is that when we do a flower dissection, we are going to be cutting these vertically. We want to cut them in half as symmetrically as we possibly can. Before we cut them in half, it's really important to take a really close look at how the flower is arranged. And the nice thing about this great big lily is that it's sturdy enough that I can kind of bend it. I can take that overhead straight at my face look, thinking about the different whorls. I can also flip it over and see where that pedicel connects in. And sometimes I may see a nectary that has some of that sugar water inside, and sometimes I may not. It all depends on the individual flowers. Some flowers make great big nectaries, and others, like this one, yeah, kind of minuscule. Doesn't really look like it exists. Sometimes looking from this angle, I can see things that help me understand my whorls. So as I move this in, what you are going to see is you are going to see two layers. You'll see these three kind of petals that are your first layer, and then you'll see petals, see these three structures like this one that are, hang on. Then you'll see the three layers like this one that are almost looking like they are on an inner layer. So we've got structures like these that look like they're on the most outside layer. And then we have structures like these that look like they are the most inside layer. I see the same thing when I flip it this way. Again, these structures look like they are my outside layer, and these structures look like they are a more inner layer. Because this flower is mature, these two views are really helping me to understand which of these are sepals and which of these are petals. Remember, our sepals will always be our outermost layer. So here we can see three sepals. These would have been green while the flower was young. They would have been very sturdy, protecting the developing internal reproductive structures. Our next layer will be our three petals. And so those three petals are true petals. They've got the majority of color, but we can see that our sepals took on color as they matured, so that now we get this cohesive, really dramatic, hey, pollinator, come here, type of structure. Now, what I love about lilies is that our stamen layer is so dramatic. Each one of these are individual stamen. We can see our dark pollen chamber. We can see that structure connecting the pollen chamber back into our flower. Now, what's missing from this flower is the carpel. Now, what happens is, for a lot of flowers, when they mature and they get older, the carpel layer deteriorates. Sometimes it is the first structure to deteriorate. But I am happy to show you that in this lily, we have a very dramatic carpel layer. This is our carpel. We can see our stigma, and our stigma has been thoroughly dusted with with pollen, which is why it has that kind of darker coloration. So we got our great big stigma, a very long style, and when we cut it open, we should be able to see our ovary and our ovule. Now I've bent this a little bit to pull it out of the whorls. So if I put it back where it was and tip it to you like you were looking straight down on it, we have our outer layer of sepal, our more inner layer of petal, all of our stamen, and our stamen are encircling that carpel layer. And just to really emphasize it in this dead and dying flower, we can see that carpel with the characteristic tip and that characteristic stigma tip is what we will be catching all of the pollen. This one is a little bit lighter, and so it didn't collect as much pollen, but hopefully it helps you to understand how a stigma-style carpel layer will look in the center of a larger, more mature flower. So we will cut our flower in half vertically, trying to make two as symmetrical as we possibly can structures. When I cut this in half, what I will be able to see is I will be able to see that long structure connecting in to the ovary. Now, unfortunately, I cannot see the ovules inside of the ovary unless I had a dissecting microscope. Now, I have a few more flowers that I want to talk about today. The lily is sort of our quintessential flower because it's fairly straightforward to identify sepal, petal, stamen, carpel. But when we get into other flowers like the crocus, it can be a little bit more confusing. And so again, if we take that straight on look, or we look from the back straight on, we can start to see those different layers. So by gently peeling back those outermost layered structures, I can see that our crocus has exactly 3 sepals, and then layered underneath of those will be 3 petals. And what I love about this crocus is that we have such a tight stamen layer. It is so tightly clustered around the carpel layer that it's almost impossible to really see the carpel inside. A lot of flowers will do this. That's kind of guaranteeing that if there are no bees around and it's not windy, that some of your pollen is going to fall on your carpel layer. It's going to fall on that stigma. It's just going to take a little bit of effort, but it will fall there and you will get some self-fertilization. Again, not the best, but it's better than no fertilization. So at least your kids will get some genetic variation, even if it's not the ideal amount. So I split the crocus in half as best as I could. And now we see the elongated pollen chamber. The elongated pollen chamber is this long yellow structure. We can see it really well right here. And then our kind of more orange structure, and pulling it off to the side, that is our carpel layer. Now again, when we look deep into the flower, it's a little challenging to see the ovules, but that kind of bump out was the ovary. Once fertilization occurs, then it will start to expand into a fruit, but the fruit is the next unit. Another flower that I would like to show you is the snowdrop flower. And I love this flower because just holding it like this, it looks like, well, these should be the petals, but they are not. Remember our whorls. These outer 3 structures are actually the sepals. And as I blow that up, we can see petals on the inside, and there's a layer of 3 very tightly clustered around the stamen and the carpel layer deep on the inside. What I like about our snowdrop is that our carpel layer is connected to a great big ovary. So finally, we can see that great big ovary by gently cutting it in half and peeling it away. We see that more traditional carpel, and we can just barely start to make out some of the little round dots of the ovules. If I had a dissecting microscope, I could really blow those up and see those in some detail. So a bit confusing initially, but if we keep those 4 whorls in mind, sepal, petal, stamen, carpel, we know that the 3 dramatic structures on the outside are actually sepals. And then we've got a very dramatic ovary with ovules that we can visibly see inside. Now, my last two flowers that I want to talk about, first being the iris. And I love irises. They are just gorgeous. They're beautiful. They're very unusual. But we have to see this as three separate structures. If we start to think about this as three separate flowers all fused together, then we can start to understand our whorls a little bit better. And the confusing thing about irises is how they grow. So initially they start out and everything is all tightly tucked together. Now based on that, and again remembering my outermost layer is going to be my whorls, I know that this really dramatically colored layer, it tends to be a deeper purple in this plant, and we have the yellow striping on it. These are actually the individual sepals. They're doing a lot of double duty here once they mature. So then, up here are my petals. And my petals have all started to fuse together, and they almost look like they're fused together throughout the entire flower structure. So the big question is why? Why would a plant do all of this fusing of sepals and petals and then hide the reproductive structures deep inside of the flower structure? And the reason why is this flower is going to really make the pollinator work hard. If you think about the insect or the bee that would be pollinating this, it would get attracted. It would go, whoa, here's my runway, zoom in, but then it's got to walk all the way back here. And by walking back here, you can see that little yellow structure at the top of my finger, it's going to get a bunch of pollen. So here the flower is guaranteeing that the pollinator will get some of the sperm, get some of the pollen. So then maybe it flies to the next flower. It crawls back in. Well, then it's going to be depositing and dropping off that flower. This plant is guaranteeing that its sperm is going to get combined with the stigma, style, ovary, ovule, egg of another plant. It's guaranteeing we're going to get some sexual reproduction to happen. So it's going to invest a lot more energy in putting all those structures together. We see this happen a lot with flowers, where the plant has started to fuse things together. And the final one that I want to talk about is a rose. Now, I know to your eyes, you're like, oh, what a beautiful flower. No, this is beautiful flowers. There are hundreds of flowers in a rose. So what we have to think about is, again, we have to think about our whorls. And if I flip it around, we can clearly see the sepals in the back. Yay! We got one whorl. Each petal is connected to one individual flower. So, if we look at all of the different petals, and instead of seeing the petals as being part of one flower, we see the petals as being part of 1, 2, 3, 4, lots, then we start to understand what this plant is doing. This plant is doing a head inflorescence. Lots of flowers. Each flower has its own sepal. Well, a lot of flowers in this head inflorescence have their own sepal. A lot of the flowers will have their own petals. They'll all have their own stamen and their own ovules, ovaries with, their own carpels with ovaries and ovules. So this plant is essentially combining lots of flowers together, lots of little individual flowers together into one great big structure. Big question is why? It comes back to pollination. If everything is all in one location, you're more likely to get all of the genetic mixing between the pollen and the eggs that you need to have happen. So, I know that initially flowers can seem quite confusing. And if you have any questions, don't hesitate to take pictures. Email me with your thoughts, your ideas of what it is that you're looking at. Keep coming back to the quintessential lily, coming back to those 4 whorls, and then looking at the flower in front of you and going, okay, how did you modify those 4 whorls? What are you doing different than the lily? What are you doing different than those 4 classic whorls and the classic picture that you've drawn in your notes from the lecture slides? email me if you get any questions.